initials at the end of my name, CBSP, RBP, and these are biosafety certifications that are recognized by the American Biological Safety Association. But there's also a very important ASM Specialist Microbiologist Biosafety Certification, and actually you have to have this one to get the CBSP. So the CBSP stands for Certified Biosafety Professional, and the RBP stands for Registered Biosafety Professional. There's just minute differences between the two. RBP is more work time on the job related certification, and the CBSP has an exam which ASM writes. And I'm actually on the board that writes that exam.
So do you have to take courses? You get the certification? Or?
The CBSP, you have to have, and now I should refer to my ASM friends, I think it's 34 credits of microbiology, or a discipline, similar discipline, and you'll have to take a board exam. It's a really, really difficult exam. Okay, so just a little bit of biosafety in the news. What do we do in biosafety? I know that everybody's very familiar with the biosafety officer, and I apologize ahead of time because in the United States we're very regulatory as far as biosafety, and I think it really hits academics very hard. I'm working in public health biosafety. We're working to stand up public health systems internationally. So I work internationally. It's much more relaxed. We don't have the regulations. We just have the guidelines to go by, and it really works out because biosafety is being done by scientists where it should be, in the lab. Biosafety in the news. Just some of the things that we work with. First of all, I'm sure you think of biosafety as inside the lab and laboratory-acquired infection. And this morbidity MMWR report speaks to a fatal laboratory-acquired infection by Yersinia pestis, and it was an attenuated strain. So, you know, things can happen, things do happen in the lab. And so there's good things about biosafety. There's good things about having that safety factor in the lab. West Nile virus spread. Unfortunately, you know, we're so focused on the laboratory. We're so focused on the containment. Here's an example of, you know, the spread of a disease, and this is a hemorrhagic or neurotrophic disease. It's really a very bad disease, can have long-term consequences to this disease. And it just spread throughout the United States. It became endemic in the bird populations, horse populations, mosquito populations, and just went to town and just spread. And we really need for scientists to be involved in biosafety. Biosafety is a preventative science, and it is a science. And so we need for good people to be thinking about this. How can we prevent this type of occurrence?
It took only 15 years to spread throughout the...
Right, right, yeah. But once it got stuck in that bird population, there was no way to really stop it. So once you have the environmental factor there, then it's really difficult to stop that kind of spread. So biosafety, what else is it? It's a discipline that crosses across the life sciences. So if you're an entomologist, if you're a microbiologist, if you're a bacteriologist, if you're, you know, no matter what ologist you are, there's some component of biosafety in your work. And it would be good to have people thinking about that instead of dreading the dreaded biosafety officer visit. This is biosafety in the news again. This is more the bloodborne pathogen diseases from the 1970s to the 1990s. We had horrible outbreaks of diseases, Ebola, hepatitis B, throughout the world. A lot of these were caused by syringes, dirty syringes. And what happened was, well, I guess it's twofold, because some of the syringes where you're old style glass syringes with the metal tips. I don't know if you've ever seen those.
Maybe too young to
In
see
museums, but in the olden days, syringes were made of glass and you had a metal needle attached to it. And these could be boiled and they could be sterilized very easily. The invention of the disposable needle, which was supposed to improve safety, and did improve safety in hospitals in developed countries. What happened when this was imported into developing countries, they needed to reuse these, and they couldn't sterilize them. And so we started to see these horrible outbreaks of disease that were bloodborne diseases. So now, in response, we have to get bloodborne pathogen training every single year. And it's a good thing because it makes us think of sharps and how we can avoid those percutaneous exposures to pathogens. Drug-resistant TB, just another biosafety problem that is part of a public health problem. And how do we keep multiple resistant bacteria from spreading? What do we do? We're preventative side, so how do we prevent spread? This is the zoonotic diseases, emerging and infectious diseases. And once again, the question is, how do these diseases go from animals? Because these are diseases that spill out of the animal population into the human population. Why do some of these diseases spill out and then just retreat, like rabies? It never really becomes an epidemic. Why do other diseases go on to become epidemics, and why do a few, like HIV, go on to become only human diseases? These are questions that impact public health, but they are biosafety questions. Any type of question that you can think of with a risk assessment involved in it is a scientific research project in biosafety. So if you're doing research in the future and you want to publish, you can publish both in your discipline and in biosafety. There are publications. Most scientists don't think about that. Okay, just a little question. What do people think? Major cause of laboratory-acquired infections.
Not doing the procedure correctly.
Right. Operator error.
Yep.
What a shock.
We all staffed, we've all staffed ourselves with a syringe and hopefully there was nothing in it.
We have to pay attention.
Yeah.
So, you can surround yourself with beautiful facility, wonderful equipment, but if you're not working it right, you're at risk. Just a little background on the history of biosafety. The history of biosafety grew out of the science of microbiology. As these scientists came on board, tissue culture, recombinant DNA, animal studies, biotechnology, the biosafety followed with the science. The biosafety was initiated by the scientists. And this is what the person who's considered the father of biosafety, Dr. Weedham, worked in Frederick at USAMRIID and developed the science of biological safety and the idea that we have layers of containment, the facility, the equipment, and the practices, and all of these are important to protect the scientists in the laboratory. Because prior, of course, biosafety initiated with the first lab infections, and people were keeping documentation of laboratory infections very, very early on. As soon as they started working with the microbes, they started keeping documentation of laboratory-acquired infections. And actually, in the late 1800s, the first cabinet, so to speak, which was a wooden box, was used for culture of microbes to prevent exposure to scientists.
This cholera lab by pipette, was it mouth pipetting?
Yeah, mouth
Of course it
pipetting.
was.
1800s mouth pipetting.
You haven't
Cholera.
had any bulbs.
Oh yeah.
No.
Yeah. I think the bulb, was a later.
Yeah,
was
that
a
bulb
later
was a great
invention.
invention.
Yeah.
Well, that's how we contaminated all our tissue culture with the Mycoplasma oralis.
Oh, really?
Now, pipetting while you're in media.
Oh, boy, does that make sense, yeah.
That's the most common tissue culture.
I never heard that.
Mycoplasma oralis.
Oh, wow.
Because they didn't have, then we put cotton stoppers in.
Yeah, sure.
See, that's where some of the
Aerated a little bit.
But it's amazing how simple that was.
Yeah.
Some of the cures are, for this is good laboratory practice.
Yes, yeah, very, very much. A lot of biosafety could be considered quality. What are you working with? Are you sure what you're working with is what you think you're working with, and are you working with it in the right way?
Yeah.
So a lot of it comes back to the science. How do we do biosafety? How do we assess the hazards that we're working with? Well, we just have this background information, and this is information on frequently, it's old information. Now I think you'll be seeing a lot of more different types of bacteria that scientists are working with. But, it's keeping tabs on laboratory acquired infections and how did people get infected? What were they doing when they got infected? So it's very important that we have this data so that we can start to say, is it aerosols? Is it contact with your hand? What's causing, what are the procedures that are most dangerous? And this is the way we do circumspect.
What's interesting, a girlfriend of mine, when she was working in industry, she worked with herpes simplex, and before she started her procedure, these safety officers came and said, run through the procedure with a fluorescent dye. I want to see all your protocols.
Right.
The herpes was everywhere in the room. It would have been everywhere in the room. And said, okay, now can we start redesigning this procedure?
Right. Yeah. We use the fluorescent dye for the managers because in our job we have this whole group of managers that have no science background who are responsible for the diplomatic side of things, and they're always invited into the laboratory. And they just go in like hoovers. Oh, anthrax lab? Here I go. And there's nobody, you tell them, don't do that. And the next thing you know, they'll be bringing you back pictures from, I've seen pictures that, just the hair stands on end. I saw a class 3 cabinet. Seriously, it had like giant supermarket bags in the holes. And it had this blue liquid substance, I think was disinfected, like perched on top of the cabinet, right? And he came back, and this manager comes back and he said, oh, look, I got the picture of the cabinet that they're using. And I'm like, that's a working cabinet. And he said, oh, no, they weren't working in this lab. Well, that disinfectant, there's no line there. That liquid is liquid. They're working in that cabinet. Okay, so this is experimentation that actually went on during World War II in USAMRIID, where they had human volunteers who were dissenters, war dissenters. They didn't want to fight, and so they volunteered to be infected with different types of bacteria, and they got pretty good ideas of infected doses of different bacteria. Yes, it actually happened. So this is pictures of Fort Detrick, and this is where they did the exposures. What's this called? The bubble or the... I forget what it's called right now, but you can see this one portal here, and people go put their faces up against the portal, and they would get whatever exposure.
Yeah.
I think this is the anthrax, where they had the class 3 mobile cabinet to bring out different pieces of equipment or whatever to decontaminate. But okay, so it's just this history start of biosafety in Fort Detrick area. And then came the regulations. So we start with the science, and then came the regulations. And one of the best, a couple of really good documents are the NIH guidelines on recombinant or synthetic nucleic acids and molecules. That's another one that's developed by scientists, for scientists, and it really spells out a quality management system for microbiological research. And then we have BMBL, which is the CDC document that explains how to do the risk assessment. Very, very good fundamental documents. And we'll be going over some of the BMBL risk assessment a little bit later. And the Bloodborne pathogen standard, which I just hope it helps you to sit through bloodborne pathogen training, to know that, you know, this was developed in response to those huge waves of bloodborne pathogens that went across the world in the 70s and 80s. Bloodborne pathogen standard speaks to universal precautions and standard precautions. Does anybody know what universal precautions are?
Any bodily fluid should be considered as potentially infectious.
Yes, any sample that you get should be considered infectious. It's more related to clinical diagnostics where you get in samples. And then the standard precautions are clinical standards for prevention of infectious spread. Really important. So you already know a lot about biosafety and how to do a risk assessment. I think a risk assessment is something that everybody does every single day. And we, you know, you look outside, it's raining, we pick up our umbrella, you cross the street, you wait for the little white guy to show up. That's a risk assessment. Been doing it your whole life, so it's nothing new. It's nothing that you need a specialist to come and teach you or to come and show you. You know how to do it. So just remember that when you have somebody coming in, the specialist, to tell you about risk. You should know your own risks. Every person working in the lab is responsible for their safety. Your teacher isn't responsible for your safety. Your biosafety officer isn't responsible for your safety. Know how to do your risk assessment in the lab. Core proficiencies in biosafety, if you're interested. Biosafety is a discipline that you have to know a lot about a lot of different things. You have to have this very broad knowledge of not only microbiology. We do engineering. I work in facility design all the time. I work in HVAC systems. I work, you know, with cabinets. We work with certification of buildings. We look at surfaces. We learn about, you know, different materials. We have to know the immunology, because we have to know about the vaccines, how are people protecting themselves, molecular biology, and then we have to be kind of cheerleaders also. So if you have a really great personality, we need you in biosafety. We need people who have a good smile and they really understand the microbes and how to do the risk assessment. So we spoke in the beginning about the certifications, and this is the ASM certification, and you know, it's the best. It's the highest that you can. It's the highest level certification that you
have
can
to have a PhD?
You have to have a certain amount of microbiology credits, and you know, I'm an immunologist, and I was like squeaking through.
Right, because
You
it's
know,
a little specialized.
It's a little bit specialized. You need that microbiology background. Yeah, but now I'm happy that I have both the, you know, I have the immunology, clinical microbiology background, because it diversifies me a little bit more. Okay, what is certification and why should I get certified? Most, and you can go on the American Biological Safety Association website or ASM, and they'll have careers or jobs, a job board. They'll all request certification. So the two types of certification that you'll see them request is the RBP and CBSP. The CBSP is based on this ASM exam. So you have to have that SM and RCM. Once again, just certification. They have all different kinds of certification also at ASM. So I know the food safety, pharmaceuticals. So no matter what career you get into, consider that as a way to move yourself forward in your job. Okay? And I think there's a lot of information on the back table.
Yes, and
For
there's
you?
the student,
Really good.
student membership. We have something from the national office that gives me discounts. If I get 4 of you to sign up at the same time, you get a discount. I have the paperwork.
And it
really is good.
Yeah, it's really, really good. And also, I'll put in a little plug for our Chesapeake Biological Safety Association. It's called CHAPSA, and we also have a GMU meeting here throughout the year. We have really great speakers. Okay, and that's all. Does anybody have any questions from this slideshow? Mostly history. Do you have a question?
You mentioned that having a PhD in immunology has helped you in the a more like broad expertise.
Mm-hmm.
What has been opened up specifically because of that kind of expertise?
I have a background in animal research, vaccine development, and so that's been really very helpful. There's not a lot of microbiologists that have that animal background.
Okay, thank you.
As animal, you mean mice and rats? Or
Yeah,
even
animal models
Yeah.
for vaccine validation.
So Booz Allen comes in as what? What do they do in the area of biosafety? How are
I'm
they
working
involved?
now. I'm not working. I am working in standing up public health systems throughout the world. So in the public health systems, you have these laboratory response networks, and you can go on the CDC website. You know, we model them after the CDC model, which is you have your zonal labs or your local labs, and then you have your regional labs, and then you have your national laboratory for diagnosis of disease. And so we're working on the laboratory network system, what is done in what lab, because that's a real problem throughout the developing world. How do we get low-cost tests that have high sensitivity and high specificity and avoid culture of really dangerous organisms at a biosafety level? Or how do we develop the biosafety so that it's strong enough? And do we need all the bells and whistles, all the facility design and engineering? What are the components that we have here in the United States? Because in the United States, we just, went to really strong, we think this will work. But there's really no data, no background that says, yes, you definitely have to have the double door entry for BSL-3. But you really don't need that, something else, some other component. So we need to know what, how can we reduce costs? How can we help these hospitals in the developing world? They're just overwhelmed with TB patients.
It requires a -80 and, and all this things that are just melted electricity. Because I had a student last year who was came from, I can't remember which country he came from, but in Africa, and he was like looking, going, we couldn't afford to do these tests.
Right.
What you're considering routine here in the United States would never.
Never.
No,
never. Yeah. And in some parts of the world, they'll consider their microbes are a national treasure. So they want to grow them and they want to save them at any cost.
It sounds like biosafety would go hand in hand with biodefense. Do some people borderline do both?
Yes. Yes, definitely. Yeah. Biodefense, as it's defined here in the United States, really, it starts from, so I wish I had a blackboard.
Well, you do. You can make this go up, and this is a whiteboard. It works.
Okay.
I promise you. And did anybody, but do we have a marker? Let me see if I have one in my, I may have one in my backpack. Yes, I have a marker.
Okay. So we have the chain of infection, right? And it's often visualized as a chain, and I'm a very poor drawer, but you can, because the idea is that any link in this chain, if it's broken, you can stop an outbreak. And the chain of infection starts from the pool, the environmental pool where the microbe is found, and it goes through the portal, exit portal, what is the vector of disease, so the vector, I'm trying to remember this from memory, so it's not going to be that good, but you can look up chain of infection. And it's
Vector or vehicle?
Vector, yeah, because
vector,
or
vector,
vehicle,
yeah, or vehicle.
water. could
And
be
then, you know, has to do with spread of disease, etc. Then you start, so if you put that, overlap that with, you know, this would be number of cases on the x-axis. You'll see that, you know, in an outbreak, you'll see sporadic here and there, 1, 2 cases, and then all of a sudden you got the outbreak, you know, and this would be the outbreak curve right here, right? Where the disease really breaks out in the population. This is the point where biodefense takes over. And you'll have things like medical countermeasures, and so you can look up the biodefense report card. And you'll see that, you know, they'll be measuring things like, do we have vaccines? Do we have the supply chain? Do we have some way to disinfect and clean up the environment? Everything that they have, all the countermeasures are after the infection breaks out, right? This is where biosafety belongs. It's preventative. So this is where we really need to have some good scientists working on the biosafety. Epidemiology does it too. But when you talk to an epidemiologist, they'll say, oh, climate change causes disease. And we're here with biosafety saying, no, you know, not all climate change is causing disease, so you have to speak to me like a scientist. You have to think, you have to talk about habitat overlap. You have to speak to me about indicator organisms. So that's what we need, is for the science to go from this theory to actual working practices that we can use to develop biosafety and biosecurity for this in public health, for preventative, breaking this chain of infection at the vector. If we had known that the Culex mosquito, which was reported to be expanding the population in the winter before the West Nile virus outbreak, if we had taken care of that Culex mosquito population, would we have West Nile virus in the United States at this point?
Well, Lyme disease too, since it's moved
Lyme
from
disease.
the deer tick. It's now in the mouse population. If it's in the
Yes.
mouse deer don't make any difference anymore, and you cannot eliminate the mouse population.
Right,
Those field mice are now
right,
we're
Right.
done. It's here to stay.
right. So that's an example of people moving,
Into the
well, first of all, people changing their environment,
Mhm.
and then people moving into these semi-forested
where the deer live.
areas where the deer are. So it's behavioral changes
Mhm. Yeah.
and, you know, changes in habitat
Mhm.
for the disease vector.
Is
So
there a
this-
shortage of,
So the biodefense report card would be what you want to look for.
is there a shortage of biosafety specialists? Would they be in demand if
There's
a person gets?
There's a lot of biosafety officers. There's almost no biosafety research. So what we really need are researchers who understand biosafety. Okay. So can we put this? Oh, sure. Got the little magic button? Yeah. Thank you.
Yeah, I don't trust it.
Okay. Okay, so we'll move on. How am I doing with time? Okay, so we have a little tabletop exercise. where you are the biosafety officer and you learn how to do the risk assessment. Okay, so here's a test, because I know you already know a lot about safety. So tell me what you see in this picture. This picture was actually sent to me by, these people were so proud. They put on everything they could think of biosafety, and they sent me this picture.
Is there a water bottle there?
Yes.
Is that a water bottle?
That might be just
Oh,
for a
that's-
sample.
Okay.
Oh, it's
Okay.
collecting.
Where's the biosafety cabinet?
Uh-huh.
Here's a biosafety cabinet.
Why are they working?
Why are these people
outside?
wearing a biosafety cabinet?
Yeah, and why are they working outside of the biosafety cabinet? Why don't they put the stuff in the biosafety cabinet?
Would you want to walk in that room
No.
even after they cleaned up? No.
No, and hopefully none of them touch those
Look at the-
refrigerator, those freezers in the back.
Yeah. Yeah. That's an old time classic. They were proud.
Oh.
What about this
Let's see.
picture?
Blocking the airflow.
Blocking the airflow.
That sash is down pretty
Mhm.
low.
Yeah,
It depends on the
it's
angle.
It's just the
Just the angle view?
it's just the angle.
Okay. That's too much stuff in there.
Yeah.
How do you work in that?
Yeah. All these are probably that high. Sharps container.
Yeah, and they should be.
So-
That's too much stuff.
They're not using their biosafety cabinet correctly, and what it does is cause the air. You have to understand the biosafety cabinet is a laminar flow. It's a sheet of air. The sheet of air goes in here at the opening of the sash, and then there's a sheet of air that comes straight down, and it splits at 14 inches, so this high off the bottom of your cabinet. So if you have something this high, what you're doing is just causing the air to convections in the air. And what's happening is the biosafety cabinet is supposed to protect both the user and the product and the environment. And if you have that going on, you're not protecting your product for certain. Probably not protecting the user either.
Also, this looks like a right-handed person, and that is set up for a left-handed person. Everything's in the wrong place.
It may be just I
Yeah.
did something with the photo. That air stream going into the cabinet is so slight. If you just puff on your hand, it's a stronger puff than the air stream. So that air stream can be broken at any time. What about this one?
Oh, I've seen that. I have seen that in many a lab. Yeah, and then they expect you to clean it up.
Here's the loops.
Yeah. Oh, they touch eukaryotic cells
Yeah.
or bacterial cells. Or
Yeah.
whatever.
Yeah.
More.
This place, I actually had to get them to listen to biosafety because of spills, trips, and falls. There was so much culture on the floor that I told them OSHA. OSHA would have to take care of this. When they heard OSHA, it was like, okay, we don't have to listen to the biosafety officer, but we'll listen to OSHA. What about this one?
What is that?
What is that? Gloves?
Those are gloves.
Where are they hanging?
I don't know if you recognize this old style. They washed them out and hung them up to dry.
So that's where you'd hang like beakers?
Yeah.
Ah,
Yeah.
oh, goody.
Yeah, it's a beaker
Are
rack.
those disposable?
Well, this is in other countries, nothing's disposable. So, LAIs happen.
What is it? What is
A
it?
lot of LAIs, laboratory acquired infections.
Ah.
They're happening today, they're happening all over the world. It really is a serious problem. Not, I think, as much in the United States, but there are some in the United States. I can show you a lot of MMWR reports.
Not that many years ago when I got my PhD, we ate in the lab.
Yeah,
We ate in the lab. Lunch was
Right.
in the lab.
right.
Yeah, me too. Lucky I was in the biochemistry lab and didn't work with anything dangerous, but
Yeah.
we ate in the lab.
Yeah.
That's not
But,
allowed
we
anymore.
can always,
Yes.
we can always,
Now that you
the
mention
idea
it.
is that we can always be improving.
Yes.
And we should always be thinking about the risks.
Yeah.
And we should consider the fact that there are a lot of people that don't have the immune system that we may have. There's a lot of people that are immunocompromised, and some people don't even know it. So the Yersinia pestis infection that I spoke about in the first slide, that was a gentleman who didn't realize that he had an excess of iron. Yersinia pestis is a bug that really looks for iron, needs iron to.
He was a good candidate.
Yeah, so he didn't know he had an immune problem, was working with an attenuated organism and caught a fatal infection. Okay, conducting the risk assessment, once again, it's easy. Get it out of the way. So what we do in biosafety is we do a hazard identification based on the risk group, and the risk group is based on the pathogen. So what is the hazard? The main hazard is a pathogen. When you're working in a lab, that's the main hazard. People will come and they'll tell you, oh, this centrifuge, oh, you got to put, no. Main hazard is a pathogen, okay? Then we look at the biosafety group level. The biosafety level is informed by the risk group. And enhancements, there are enhancements to biosafety then that you can do with the biosafety level. So the biosafety level is described, we have great description by the CDC and BMBL. It'll tell you exactly what are the practices. You have to use gloves, you don't have to use gloves. But then you can enhance. Oh, I think I need a respirator because this is a microbe that has airborne, can affect you by airborne.
4 is the highest level?
4 is the highest level, yeah. So 1 is teaching labs. 2, we work with, we'll go over this, I guess, here, right here, the risk groups. So what about the pathogen determines the risk? I just want you to take a minute and look at these risk groups, especially start from risk group 4, and think about what are 3 elements that they're calling out here in the BMBL that indicates whether or not your pathogen is a higher risk.
Respiratory, it's a respiratory.
No, they don't. There's no respiratory in here.
Do we have a vaccine for it?
Therapeutics.
A therapeutic. Okay.
Available therapeutics.
There isn't one.
Yeah. What else?
How likely it is to cause
Disease. Yeah, disease.
And is it a lethal disease or, just a chronic infection versus you get Ebola, you're
Transmission.
Usually they call out transmission, number 3. So 3 things. Disease causes disease, whether or not there are available therapeutics, and transmission. How does it spread? Does it spread rapidly through a population? Doesn't have to be aerosol. HIV was not aerosol.
But HIV is only classified as 2, isn't it?
It depends if you're working with
That's concentration.
with the concentration and also with live.
Yeah. So that just to underline community risk. Those are the 3 things in the hazard. So the risk group informs the biosafety level, but you can go up or down. And often researchers look for a way to go down. They attenuate their organism. They're working with smaller numbers of bacteria or virus. So you can go down by adding safety measures. You can go down. Vaccination is one way in other parts of the world that they decrease the risk. So BSL is defined by the containment layers. In the BMBL, you'll see they'll talk about facility engineering and practices necessary to contain and protect you. The risk group is defined by the pathogen.
Question.
Yes?
Are these standardized throughout the entire world? These BSL levels and risk groups and everything?
The WHO, the World Health Organization, has a document that's recognized by most countries internationally. However, all of this in biosafety, we say you can't, you have to be able to decide what the risk is yourself. In your facility, for your specific immune system, what is your risk? You have to be able to determine the risk and apply measures as appropriate. So if you're on antibiotics, you could use a little bit more PPE, for instance, that day, just because you feel like you have higher risk. In some parts of the world, in countries where the states control everything, they don't allow the risk assessment. They want to be able to, at the federal government level, define what the working procedures and practices should be in the lab. So, for the most part, it's accepted throughout the world. It is the way to protect yourself. But there are guidelines, but they aren't regulations. Okay, so once again, BSL can be decreased based on decreased risk, and these are some of the ways that we can decrease the need for higher BSL, and BSL can be increased if we think that aerosols, the reason we call out aerosols is because of the potential to infect more people. Yeah. Well, I mean, you drop a plate in the lab, the aerosols will infect everybody in that lab. So higher concentrations of organisms, these would all be indicate a need to increase risk. In biosafety, okay, so once again, we use this facility, engineering, and safe practices to decide the biosafety or to define the biosafety level, and then we layer it. So one of the very common mistakes I've seen people do is they walk into a lab and they say, oh, there's BSL-2 lab. Yep, we got the sink, we got the, the surfaces, BSL-2 lab. If the working practices aren't BSL-2, if people are in that lab working with, a highly infectious organism, this is not a BSL-2 lab. So it has to be facility, engineering, and practices, and we have to layer them. Hierarchy of risk control, elimination being the best. If you can eliminate all the way down, and the very last one we have is PPE. So SOPs and PPE, or training and PPE, considered the least effective. Why?
Because every person has different practices.
Every person has different practices, and remember the first slide where we said operator error is the results in the most LAIs. So we don't want somebody putting on their PPE wrong and getting sick.
Or forgetting to take it off when they leave the lab.
Or again, yeah, another big problem. Okay, so let's first go through a hazard analysis. If I could just, can I turn this off, blanket out?
I don't know if
You don't know about that?
You just want me to turn off the projector?
Nah,
Yeah, it
well,
just-
but it takes so long to turn back
Don't
on.
worry, don't
Don't
worry.
turn it off.
Don't worry
There
about it.
should be a
Let's go
net.
through a hazard analysis first. If people have time, it's 7:24.
We have time? You guys
have
okay?
time?
You were going back to study for my exam. Look at that groaning.
It's gonna be an all-nighter anyway.
An all-nighter. Okay, let's just go. Let's try just one hazard analysis. We'll look at Yersinia pestis. Or let's look at the hantavirus. Hantavirus.
I always love Yersinia pestis. I tell them
Yeah, I
when we do our
love
intro
Yersinia.
twos that if they get that as the organism, I promise you I will give it to them.
Thank you.
Look at the hantavirus.
Thank you.
So remember that we're doing a hazard identification. What are the three things about the pathogen we're thinking about?
Transmission, how virulent
It
is
causes
it?
a disease.
Disease.
And how is it transmitted?
How is it transmitted?
And is there a vaccine or something?
Is there a vaccine?
Do we have
Okay, so knowing that, why don't you, somebody suggest a risk group for hantavirus. And I'll try to quickly find hantavirus in the BMBL and see what they suggest. Okay, pathogenicity. Who wants to speak about pathogenicity? Hantavirus. Does it cause disease?
Yes.
Yes.
And it says severe and often fatal.
Severe and often fatal.
So it's
Says
3 to, where you should start at 3 and work our way up.
probably we should start at 3. Are there therapeutics for hantavirus?
No.
No, viruses we don't have really good therapeutics, right?
So that should move it towards a 4.
We're
Getting closer to 4.
well, an unknown.
Yeah,
Yeah.
it's always a 4.
Yeah. Yeah.
And also some is aerosolized.
So it's
It's
transmission.
aerosolized. So the BMBL says BSL-2 practices, containment equipment, and facilities are recommended for laboratory handling from sera. So now they're separating out clinical samples from cultures.
Right.
Okay? Sera from persons potentially infected with hantavirus. Use of a certified BSC is recommended. BSC is a biosafety cabinet. Potentially infected tissues should be handled in BSL-2 facilities following BSL-3 practices and procedures. So remember we talked about you can move up in your BSL or down in the BSL depending on the risks. Experimentally infected rodents, ABSL-2, serum samples from potentially infected rodents, BSL-3. So, you know, they move around the biosafety level. The risk group does not determine, but you were very correct to say risk group 3 organism. It's a very dangerous organism to work with, but the biosafety level then depends on how you're working with it, what you're using to work with it. So that's how you do the hazard identification. Now we're going to do a risk assessment. The risk assessment, you're going to look at the risk and then decide risk mitigation methods. What would you do to mitigate risk? And this is a multi-state outbreak of Salmonella typhimurium. I think it was in 2011, the CDC decided that, or found out that there were multiple cases of teaching lab strain of Salmonella, that people were getting infected, and actually many took home to their children. So they had multiple outbreaks. And so then they went through, here's the epidemiology. You can see that it's just a few here and there all across the school year. That's the epidemiological curve. Try to think of how you would mitigate the risks to this organism in a teaching lab. Make a suggestion and then we'll talk about what the CDC found.
What do you mean by mitigate the risk?
Decrease the
To
chance.
decrease the chance that the students will become infected.
Make sure.
Use a different strain.
They could, but this is a teaching strain. It's low infectivity, but it seems like, you know, somebody
Well, we have to
took
make,
out enough.
we have to make sure that everybody before they
Wash
leave
their
the lab
hands.
must wash their hands.
Wash their hands. That's how they're getting infected. It's not aerosolized. It's on their hands
So make sure.
or on their lab coat. They're
Right.
walking their lab coat out.
Mhm.
What else?
Well, the equipment that, you know, like if I have a pen, I put it down on the lab bench,
Yeah,
and then I
yeah,
put it in my mouth, which I don't
yeah.
never do.
Disinfection of surfaces before you leave the lab.
Also, your opening doors had to have one glove only on and one glove off, like
Yes.
you do at Prince-William.
Yeah, don't use gloves on common surfaces. Very good. Yeah. So, you know, just common sense things. But some of these people were not working with the organism that got the infection.
Right.
They walked into a lab, they thought it was clean, it looked clean.
Right. You know, you open up probably that refrigerator door handle was the worst thing, because somebody goes to put the culture away and never, didn't think that somebody could come by with it without a glove on or.
So what did the CDC recommend? They actually found, wash your hands is a big one, but they actually found that signs and symptoms of the disease was a big one. Posting, hazard communication, posting what is in this lab and what it causes. And if you have these symptoms and you've been in this lab, think about it. Don't take it home to your kids. Another big one that they're starting to look at is cell phones. Because people are using the cell phones in the lab, then they wash their hands going out of the lab,
And you hit
then they grab their cell phone again.
That's
Yeah.
sliding with your finger.
Yeah, so no cell phones in the
No
lab.
cell phones in labs. Yes! Oh, yay!
I'm
Yay, I love that. Thank you.
Biosafety
Especially not
awareness.
organic chemistry.
You know, so it doesn't have to be rocket science. It doesn't have to be, some type of engineering change. It's mostly behavioral changes. Okay, and that's all we got.
Well, we bring backpacks.
So
into
I'm
lab,
really
of
sorry I
course.
went a couple minutes over.
That's fine. This
And
was
I know
great.
you got tests, and I won't be offended if you didn't need to run.
Thank you.
If anybody has any questions.